And chromosomal segregation involves many molecular machines, including the centrosome, the cuticle, and the mitotic spindle. And today I'll talk about our structural mechanistic studies of the anaphase protein complex, or APC. And the APC is a large multi-subunit complex which regulates progression through the cell cycle by mediating degradation of cell cycle proteins via the ubiquitin proteasome system. And the APC was discovered actually by Aviv in 1995 in his studies to understand how cyclin degradation is regulated. And Aviv and his colleague Aaron Shkolnisky also discovered protein-mediated ubiquitination, or ubiquitin-mediated protein degradation. So his research has really made a big impact on my research and inspired my research. So the APC is best understood for its function in controlling chromosome segregation in mitosis, and this occurs at the metaphase-anaphase transition. So at metaphase, all the chromosomes are aligned at the center of the mitotic spindle at the metaphase plate. And the bipolar cystochromatid pairs are arranged so that both centromeres of the cystochromatid are attached to mitotic spindles from opposite poles of the cell. And this cystochromatid is then under cohesion because of this cohesion ring which encircles the two cystochromatids. And on the answer of anaphase, the cystochromatids are separated because the cohesion ring is disassembled. And the APC regulates this transition from metaphase to anaphase. And this video here shows the events of the cell cycle and mitosis. And during the cell cycle and mitosis, the cell undergoes quite profound biochemical, structural, and morphological changes. And the chromosomes are segregated in a very synchronous way. So that ensures that both daughter cells receive the same number of chromosomes. And the synchronous segregation of chromosomes is mediated by checkpoints. And the checkpoints, in turn, control the APC to make sure that the chromosomes segregate at the right time. So defined proteolysis controlled by the APC regulates the metaphase-anaphase transition. So in mitosis, the APC is activated by phosphorylation on multiple sites, and also by the binding of a co-activated subject called CC20 to generate the APC CC20 complex. And this complex then recognizes and ubiquitinates two proteins, cyclin B and securin, and these two proteins are inhibitors of separase, so their degradation by the proteasome system leads to the activation of separase, which then cleaves the ring, cohesion ring, which maintains cystochromatid cohesion. And to do this, the APC binds its substrates through these short degron motifs, or destruction motifs, or short linear motifs, which Madame Baboon introduced this morning. And CT20 recognizes three types of degrons, the so-called D-box, the Abba motif, and the Ken box. And this brings this substrate in close proximity to the catalytic site, which is formed from an APC 11 subunit, which is a ring subunit, which binds the E2 ubiquitin conjugate to transfer ubiquitin onto lysines of the substrate. Now, today I'll describe two studies. The first study I'll describe is to explain how the APC is controlled by phosphorylation in mitosis. Then I'll spend most of my time describing how the APC is controlled by this bimosome checkpoint, which is a checkpoint which determines that all chromosomes segregate simultaneously at metaphase. So I'll explain first how the APC is inhibited by the spinosome checkpoint, or SAC, and then explain how the APC is reactivated when the SAC is signed when all chromosomes are properly attached to the mitotic spindle. So the SAC coordinates the activity of the APC with the complete and correct attachment of all cuticles to the mitotic spindle. And cuticles are large protein complexes which assemble onto centromeres in mitosis. And detached cuticles activate the SAC to inhibit the APC. Now this is achieved because the spinosome checkpoint, or SAC, generates a complex called the mitotic checkpoint complex, which binds to and inhibits the APC CC20 complex. So before I describe how the APC actually recognizes substrates and ubiquitinates them, I just describe the overall structure of the APC. So the APC is a protein complex of 15 different proteins. We know from work which we did in collaboration with Carol Robinson's group in Oxford that many subunits are present as 2 copies per complex. So the total mass of the APC is about 1.3 megadaltons. The catalytic subunits are composed of a ring subunit APC11 and a cadmium subunit APC2. And substrate recognition, Begram recognition, is mediated via the co-activator, either CT20 or CH1. And the APC uses two different co-activators. And switching between these two co-activators allows the APC to recognize different substrates during different phases of cell cycle to allow different cell cycle phases to be controlled by the APC. But together, the catalytic module and the Begram recognition subunits account for only 15% of the mass of the APC. So 85% of mass APC is composed of these 7 large structural subunits. And many of these contain multiple repeat motif sequences, such as the TPR repeat, which actually Gideon described this morning. In fact, the TPR was first discovered in APC subunits about 1994. And then the APC1, the largest subunit, contains WD40 repeat at the N-terminus and also a PC repeat domain in the middle of the protein. And these large scaffolding proteins function to coordinate the catalytic module and the Begram recognition module. So in early mitosis, the APC becomes hyperphosphorylated, and the two subunits which are most phosphorylated are APC3 and APC1. On APC3, there's a large loop insertion within the TPR domain, which becomes hyperphosphorylated. With an APC1 loop, we call the 300th loop within the W40 repeat domain, the N-terminus also becomes hyperphosphorylated. And the phosphorylation of these sites allows the APC to bind CT20, which then activates the APC to ubiquitinate substrates. So, sorry, I went too fast there. So, to understand the structure of the APC and to understand how it's regulated and how it recognizes substrates and how it ubiquitinates substrates, we needed to obtain large amounts of material for structural and biochemical studies. And the APC has a very low native abundance. So to obtain enough material for these studies, we expressed the APC in the insect cell baccalaureate expression system, where we could obtain about 1,000-fold high levels of protein compared to the endogenous levels of the APC purified from either human cells or yeast cells. And this just shows 2 STS page shells of 2 functional APC complexes. And then we determined the structure of the APC using cryo-electron microscopy, and this shows a cryo-electron micrograph of the recombinant APC in complex with a CH1 co-activator and an inhibitor called EMI1. And these data were collected on a Polara electron microscope with a Falcon 2 detector in about 2014. And in these data, we could obtain a 3.6 angstrom structure overall of the APC. And, but this shows the local resolution map for the APC, and it shows that some regions, which are the central regions of the APC, are much higher resolution than the more rigid regions, whereas some of the more peripheral regions are more flexible, and these turn out to be the interesting areas, which are the substrate recognition module and the cadmium module. But overall, the APC EM map had high resolution, and we could actually fit side chains to the EM density. The model was built overall by fitting structures of different subunits determined by crystallography, and also ab initio building of some subunits we could not crystallize because they were unstructured proteins but become structured and binding to the APC, such as this APC15 subunit here. Then more recently, we collected data on a Cryos Titan electron microscope using a Falcon 3 detector in electron counting mode. And also we used the multi-body refinement routine in Relion, and this allowed us to obtain a more high-resolution structure of the APC to 3 angstrom resolution overall. But this also allowed us to define some parts of the structure which are less well defined in the earlier map, including regions of the APC from APC12, which is predicted to be unstructured, but actually, in fact, in the context of the APC, forms its interface between APC1 and APC2. So this is an example of some of the unstructured proteins which actually are important in mediating inter-subunit interaction to stabilize complexes. So this then shows the overall architecture of the complex. It forms this triangular-shaped molecule, and it features this central cavity where most of the activity of the APC occurs. And then the substrate recognition module composed of the co-activator and the APC10 subunit, which is also part of the substrate recognition module, is shown here at the top of the cavity, whereas the catalytic module is at the bottom of the cavity, and it's composed of APC11 and APC2. So the TPR proteins I mentioned earlier actually are paralogs, and the 4 homologous TPR proteins all form homodimers, and they form this TPR lobe on one side of the complex, and these 4 TPR proteins stack in parallel to create this pseudo-symmetric structure. The platform region here is composed of APC1, APC4, and APC5. So then this shows how the APC recognizes the 3 degrons. So these are the D-box, Chem-box, and Abimotif. And this work was actually done by X-ray crystallography by crystallizing the coactivator in the presence of the degron sequences. And these bind to their cognate binding sites on the coactivator. But the D-box actually binds to a co-receptor formed by the coactivator and the APC10 site unit. And then this places the substrates close to the catalytic module, which then binds to the E2, in this case UPCH10, which is also discovered by Avram Hershko. It's been the major E2 for APC activity. And then this places the lysines of the substrate in close proximity to the charged E2 ubiquitin conjugate, which allows the transfer of ubiquitin onto the lysine substrates. And then it was shown by Brenda Schumann, actually, that APC2 contains the C-terminal WHB domain, which is important in binding to the backside of UPCH10. And this defines the specificity of the APC for UPCH10, because most other E2s don't require this secondary interaction between a cutting subunit and the E2. Most E2s are recognized by the ring domain, and the ring domain actually functions to bind to the E2, but also to bind to ubiquitin and promote this so-called E2 ubiquitin closed conformation, which is more active than the open conformation. So ring domains both bind to the E2, but also induce this closed E2 ubiquitin conformation to activate the intrinsic activity of the E2 ubiquitin conjugate. But the WHB domain is also important in actually stabilizing the closed conformation of the UPCH10 ubiquitin conjugate. Now, the coactivator, as I mentioned, is primarily important in recruiting substrate to the APC. But also the coactivator is actually an activator because it induces the conformation change of the catalytic module from a down conformation in the apo state to an up conformation in the complex of the coactivator. And this up conformation of the catalytic module of APC 11 and 2 allows the binding of the E2 to the catalytic module, because in the down state, in the absence of the coactivator, the binding site for the E2 is blocked by the platform subunits. So I'll just now briefly explain how the APC is regulated by phosphorylation. And this is work also by Avram Hershko, who showed in 1996 that in early mitosis the APC is phosphorylated, and this is required for APC activity. And it's later shown that this is specific for the CT20 coactivator and not for the CH1 coactivator, which is active independent of phosphorylation. And the CDK kinase and the polar kinase both are involved in phosphorylating the APC to allow activation by CT20. So the coactivators have this structure here with a C-terminal W40 domain which binds to the degrons, and then they have the sequence at the end terminus called the C-box, which mediates binding to the APC8 subunit, and a sequence at the C terminus called the IR2, which mediates binding to the APC3 subunit. And APC8 and APC3 are both TPR subunits and both paralogs. And then this, again, it shows the structure of the APC. This is actually another phosphorylated APC-CTC20 complex. And it shows that the C-box at the end terminus binds within a groove of the TPR helix of APC8, whereas the IR-tail at the C terminus binds in a similar shaped groove within the APC3 TPR helix. And these actually sites are actually homologous, and these motifs actually have a similar sequence. Now, we want to understand how phosphorylation stimulates the activity of the APC by CT20, and this is due to actually enhancing the binding or the affinity of APC for CT20. So to address this question, we compared the structures of phosphorylated APC CT20 with unphosphorylated APC CH1, because CH1 does not require phosphorylation for binding to the APC. And then we also compared the structures of the APO APC in both the phosphorylated and the unphosphorylated states. And what we found was that if you look at the phosphorylated APC CT20 structure, we see no EM density for any of the phosphates, even though there are about 100 phosphate sites on the APC. These are not visible in the EM density map. So this showed that phosphorylation does not directly stabilize the binding of CT20 to the APC, or does not indirectly via an allosteric composition change of the APC. So then when we looked at the structure of the APO APC in the phosphorylated and unphosphorylated states, we also found they have very similar structures, except for some additional density present in the unphosphorylated APC, which is absent from the absent from the phosphorylated APO-APC. So this again shows the C-box binding pocket in the APC CC20 complex, which is phosphorylated. And this pocket here is now occupied by additional density in the unphosphorylated APO-APC structure, but not in the phosphorylated structure. And the proximity of this C-box binding site to the WD-40 domain of APC1 and to this very long loop called the 300 loop suggested that this density might be a region of the 300 loop, which could then bind to this site and block the binding of the co-activator. And when we deleted this loop, we found that this binding site, this pocket now, was devoid of EM density. So that suggested that this density, which you couldn't determine its sequence very well early on in this reconstruction, suggested it was density arising from residues of the 300 loop from the APC1 subunit. And then if we then delete this, this loop, we actually constitutively activate the APC without requiring to phosphorylate it. So this now is comparing the activity of the APC without phosphorylation. When we phosphorylate APC, we can stimulate the ability of APC to ubiquinate substrate, in this case cyclin A. If we just delete this loop, we can actually activate the APC even without phosphorylation, and we don't really increase the activity of the APC by phosphorylation. So that shows that phosphorylation simply removes this loop from the C-box binding pocket. And then if we look at this EM density in more detail, we see that the region of the so-called APC1 autoinhibitory segment of the 300 loop actually has sequence similarity to the C-box sequence. And so this autoinhibitory sequence actually structurally mimics the structure of the C-box bound to the C-box binding site. And the sites of phosphorylation which are required to activate the APC are flanking this region which mimics the C-box binding site of the co-activator. So by phosphorylating these residues surrounding this central region, which contains this arginine and phenylalanine residue, the additional bulky phosphates would actually cause this autoinhibitory segment to be displaced from the C-box binding pocket and then allow the binding of the C-box in this site to activate the APC. So moving now to the question of how the APC is controlled by the spin assembly checkpoint or the SAC. So as I mentioned, the SAC is activated by unattached catechol. So a single unattached catechol in a cell which contains 46 chromosomes is sufficient to inhibit the APC activity and to prevent anaphase onset. And this unattached catechols inhibit the APC by mediating the assembly of another complex called the mitotic checkpoint complex, which binds to the APC CC20 complex to repress the activity of the APC towards securin and cyclin B, therefore delaying anaphase onset. And this delay persists until all catechols attach correctly to the mitotic spindle. So this is now a crystal structure of the MCC from fission yeast we determined a few years ago. And this shows the triangular shaped structure of the MCC, which is in this case lacking BUB3, because BUB3 is not required for the checkpoint in fission yeast. And this complex showed that this complex is composed of CC20 and then the MAD2 subunit and a BUB1 subunit. And the interaction of CC20 within this MCC complex suggested early on that the mechanism of inhibition of the APC by this formation of the MCC was due to sequestration of the CC20 away from APC, because CC20 is the activator of the APC, but it's now present in this inhibitor. But it turns out that's not the case. It turns out this MCC subunit complex interacts with APC CC20. So it's rather a paradox that the inhibitor itself contains the activator of the APC. But the assembly of this MCC complex requires a conformational change of the MAD2 subunit, shown here. So in the inactive state of MAD2, called OMAD2, this state doesn't bind to CC20. And on activation of MAD2 to the so-called closed state, which can bind to CC20 and then bind to BUB1 to join the MCC, there's a large reconfiguration of the central beta sheet occurring. This is work from Hongtao Yu and Andrea Masaccio. And they showed that on conversion from the inactive OMAD2 to the active closed MAD2, the N-terminal beta strand is displaced from its pairing with the beta 5 beta strand and forms the first terminal of a helix called the alpha 5 helix. And then this C-terminal beta hairpin is displaced from this side of the beta sheet and exchanges for the beta 1 strand. So this exchange, or this repositioning of the C-terminal beta hairpin creates a binding site for the end terminus of CC20 called the safety belt. And that then allows CC20 to bind with very high affinity to MAD2. And this interconversion between the open MAD2 to closed MAD2 takes about 7 hours in solution in vitro. But unattached catechores can catalyze this in a few seconds. So something's happening on the catechore which allows this catalytic conversion from the open MAD2 to closed MAD2, which is then the precursor for the formation of the MCC, which is an inhibitor of the APC. So to understand how the MCC inhibits the APC, we reconstituted the APC-MCC complex, and this involves forming the APC-CC20 complex, which is phosphorylated, and binding to it the MCC, and this is now the APC-MCC complex. And this assay shows that the APC is active without the MCC, but we add the MCC and we then inhibit the ability of the APC to ubiquitinate stachyrin, which is one of the substrates of APC-CC20. And then this shows a cryo-electron micrograph of the APC-MCC, and we determined the structure to 4.2 angstrom resolution in 2016. But more recently, Claudio Ferri, who's actually a postdoc who worked on this project, again using the new version of Reliant, the multipolarized environment now has a 3.8 angstrom structure resolution. So then this then shows the EM density map for the APC-MCC complex, and it shows that there are two CC20 subunits bound to this APC-MCC complex. So one is the CC20 of the APC, and the other one is the CC20 of the MCC. And this shows the MCC bound adjacent to the co-activator and also adjacent to the catalytic module of APC2, APC11. And sorry, I should actually. So this just shows the movie which indicates the overall conformation of the APC-MCC structure. It shows how the AMC is located within the central cavity of the APC. And so the binding of the, the binding of the MCC to APC-CC20 induces the conformation change of the CC20 of the APC, so it tilts by about 90 degrees and moves away from the APC10, which is the co-receptor site for the D box. And then the ring domain APC11 also shifts upwards, and the WHB domain of APC2 becomes ordered, which now interacts with the TPR domain of BABA1. So the key subunit in the MCC which inhibits the APC is actually the BABA1 subunit. And this is composed of an N-terminal TPR domain, and then a central unstructured region which contains 5 degrons. So in total, BABA1 contains 2 copies of each of the 3 degrons. So there are 6 degrons in total. And these 6 degrons bind to the 3 degron binding sites on both co-activators. So both co-activators have their 3 degron binding sites completely occluded by the pseudo-degron sites of the BABA1. So the N-terminal chem box of BABA1 interacts with the chem box binding site of the CC20 of the MCC, and then the TPR domain is formed here. But then the central region contains 5 more degrons, and the subsequent D-box, abomotif, chem box all interact with the CC20 of the APC that on this rather strange layer-like structure which basically wraps around and behind the co-activator. And then the final 5th and 6th degrons, the second abomotif and the second D-box, interact with the abomotif pocket and the D-box pocket of the MCC CC20. So all 6 degron sites of both co-activators are blocked, so that completely blocks the ability of the APC to recognize securin and cyclin B. So as well as inhibiting degron recognition, the MCC also inhibits the intrinsic activity of the APC because the WHB domain and APC11, which is the ring domain, are also in contact with the MCC. So that blocks the ability of APC to recognize, to bind the E2. So we call this conformation of the APC-MCC the closed conformation because the MCC is actually interacting with the catalytic module to close sites on the catalytic module. But using 3D classification in Reliant, we identified a small proportion of the APC-MCC particles to belonging to a second class, which we call the open state. And in this state, the MCC module is rotated away from the catalytic module, now exposing the binding sites on APC 11 and APC 2 for the E2. And this actually represents only about 10% of the total APC population in our picked particles. And this transition between the closed state to open state is accompanied by a order to disorder transition of this APC 15 subunit. Well, so in the open state, APC 15 forms an ordered helix, but in the closed state, this helix becomes disordered. And if we delete APC 15 from APC, the APC C20 is perfectly active towards securing and cycling B, and we find that this deletion of APC 15 to the APC locks the APC in this so-called closed state. So this suggested to us that the open state of the APC, which is shown here, could still bind E2 ubiquitin. So then we determine the structure of the APC complex with the UPCH10 ubiquitin conjugate, and this is shown here. So now when we add UPCH10 ubiquitin to the APC-MCC complex, we find now that the E2 can bind to the APC11, APC2 catalytic module, and this forces the MCC into this open conformation. And the EM density showed that the C-terminus of the CC20 of the MCC now is engaged at the catalytic site of the E2. And the two lysines at the C-terminus of the CC20 of the MCC, lysine K485 and K490, are now actually in close proximity to the active site. And we could show that in the context of the MCC, the APC will ubiquitinate these two lysines only on the CC20 of the MCC. So this is shown here. If we mutate these two lysines in the MCC CC20, we can actually abolish ubiquitination of the CC20. So this shows that the MCC actually allows the auto-ubiquitination of these two lysines on the CC20 of the MCC. This ubiquitination is dependent upon APC 15, which I mentioned earlier is required to open this APC-MCC complex. If we delete APC 15, we completely abolish the ability of APC to ubiquitinate CC20. So this auto-ubiquitination of the MCC is important because it actually explains how the APC and MCC are reciprocally regulated during the SAC. So during the spin assembly checkpoint, the MCC is assembled by unattached catechors, which suppresses the APC from ubiquitinating cyclin B and securin, which then inhibits anaphase onset. But in a competing process, where the APC-MCC adopts its open conformation, which allows then the ubiquitination of the CC20 of the MCC because of the binding of the E2 ubiquitin to the catalytic module of the APC, this ubiquitination of CC20 of the MCC actually promotes the dissociation of the MCC from the APC. And this occurs both because ubiquitin itself actually drives the destabilization of the MCC from the APC C20, but also the ubiquitinated C20 is also a target for proteasome-mediated degradation. So as long as the SAC is active, the MCC is then regenerated, reformed to then reform the APC-MCC, which then again represses cyclin B and securin ubiquitination. But as soon as the catechol is attached to the mitotic spindle, the SAC is turned off, and the MCC assembly is now also turned off. So that means then that this reactivation mechanism will now dominate because there's no more MCC being generated by the unattached catechors. And now that the APC will now spontaneously reactivate to form the active APC CC20 complex. So this explains actually why the signal from the SAC only occurs from the unattached catechord, because they're producing the MCC which inhibits the APC. Attached catechords do not need to generate and transmit a signal to the APC to turn on the APC. So that explains why a single unattached catechord out of 46 can actually dominate the checkpoint and delay anaphase onset, because a single catechord will still generate the MCC, whereas the 45 attached catechords are not producing a signal, and then the final attachment of the last catechord will then cease MCC generation and allow the spontaneous activation of the APC. So in the last sort of 10 minutes or so, I want to also just talk about another mechanism by which the SAC is silenced and the APC is reactivated in mitosis. So I described how MCC ubiquitination can activate the APC due to disassembly of the APC-MCC complex. But a second mechanism of Sachs silencing is due to disassembling of the free MCC. And this is what actually Xiaodong Sang sort of mentioned this morning, that this is mediated via AAA plus ATPase called TRP13, which actually is involved in the disassembly of the free MCC to generate open MAD2. And this is also actually work from Alfred Hershko, who actually discovered that the P31 comet and TRP13 can reactivate the APC after a checkpoint arrest. And he also showed that this is an ATP-dependent process. So what happens here is that the MCC is disassembled to its constituent components, and the closed MAD2 conformation is converted to its open, inactive conformation. And this process is catalyzed by TRP13, AAA plus ATPase, which requires ATP, and mediated by this adaptive protein called P31 comet, which actually brings the MAD2 to the TRP13, AAA plus ATPase. So the P31 comet actually binds only binds to closed MAD2, and it binds to the same site on closed MAD2 as BubBa1 binds. So by binding of P31 comet to CMAD2, BubBa1 will be displaced. And this brings then this complex of MAD2 still bound to CT20 and P31 comets then brought to TRP13 by P31 comet, and then by hydrolyzing ATP, TRP13 can remodel this closed MAD2 to this open MAD2 conformation. So to understand the mechanism by which TRP13 combined with P31 comet can drive this closed to open MAD2 conformational change, we assembled the complex of TRP13, P31 comet, CMAD2 and CT20, and we used a Walker B mutant of TRP13, which reduced the activity of the protein, and we also used ATP gamma S to trap this complex. And this is work by Claudio Alfieri, and he determined structure to about 4.0 angstrom resolution. And this shows the structure of the hexameric ring of TRP13 alone without the P31 comet MAD2 bound to it. And this is a homohexamer with 6 identical subunits labeled A to F. And this hexamer forms this central pore in the middle of the protein. And 5 subunits bind ATP gamma S and have a very similar conformation, and they form this spiral structure from A to E, so A at the top and E at the bottom of this spiral. And the 6th subunit, F subunit, actually is in the inactive conformation without nucleotide bound, and it bridges the A and the E subunits. Now, P31 comet then brings MAD2 to TRP13. So P31 comet binds to a site on the ring using the D and the E subunits. And it positions the MAD2 precisely so the end terminus of the MAD2 actually then enters the pore, the center of the hexamer. And then, the regions which are remodeled on conversion from the closed to open MAD2 do not directly interact with TRP13. So then this is now more detail about how the end terminus of MAD2 engages the pore loops of the hexamer of TRP13, and 7 of the end-terminal residues of MAD2 engage the pore loops of TRP13, and these are pore loops 1 to 2 of subunits A to D, and E and F do not contact MAD2 at this stage. So the process by which ATP hydrolysis by TRP13 remodels MAD2 is actually a very simple mechanism. The model is that ATP hydrolysis by TRP13 pulls the end terminus of MAD2 through the pore. And because the end terminus of MAD2 is connected to the first turn of the alpha helix, this would actually locally reconfigure the structure of the helix and also the beta sheet. So the idea is that if you, by pulling on the end terminus of MAD2, by putting it through the core of the TRP13 AAA plus ATPase, this would unwind the first turn of the alpha helix. And this first turn of alpha helix actually stabilizes the closed state because the serine residue on alpha A helix forms 2 hydrogen bonds to 2 residues at the turn of the beta hairpin. So the serine residue, serine 16, hydrogen bonds to serine 188 and histidine 191. So by simply putting on the N terminus, this would unwind this first turn of helix, disrupt the hydrogen bond between serine 16 and serine 188 and histidine 191. That will destabilize this beta-hairpin interaction on this side of the beta sheet, which will then allow it to be displaced to the other side of the beta sheet, where now these two residues, serine 188 and histidine 191, are well away from serine 16. And now the unwound helix will then form this beta sheet to then pair with the beta 5 strand at the edge of the beta sheet. And then this beta hairpin then can reconfigure to the other side of the beta sheet. And it's been shown by Kevin Corbett and Hong Tao Yu that mutation of serine 16 or mutation of serine 188 is sufficient to destabilize the closed state in favor of the open state. So this simple mechanism of pulling on the N-terminus would account with biochemical data which shows that this hydrogen bond network is important for stabilizing the closed state of the MCC. So just to summarize and just to provide some future outlook comments. So as I mentioned, the APC is inhibited by the MCC through pseudo-substrate motifs on bubble which block degram recognition on CD20. And this interaction of the MCC also occludes the catalytic module to prevent E2 binding, but the spontaneous reactivation of the APC can occur because of the open-closed equilibrium of the APMCC, which allows the binding of the E2, which then catalyzes the ubiquitination of the CD20, which then destabilizes the APC-MCC to allow APC-C20 to be regenerated. But by a second mechanism, free MCC can be disassembled by this TRP13 PC81 comment mechanism, which I just explained. I didn't explain to you that cyclin A, which is a subset of APC-C20, can resist the SAC inhibition. So this cyclin A is actually degraded about half an hour earlier than cyclin B is during mitosis. And this is because cyclin A can actually bind to the APC-MCC, and it contains additional degrons, such as the motif, which allows it to have high affinity for the APC-MCC compared to cyclin B and securin, so allowing cyclin A to be degraded early on in mitosis. And eventually, we'd like to understand how the catechol itself actually catalyzes the assembly of the mitotic checkpoint complex, and how it catalyzes this open to closed conversion of the MAD2 homodomain protein. And this requires obviously structural studies of the catechol bound to microtubules and bound to centromeric chromatin. So I'll finish here by acknowledging my colleagues. So the work I described was the work of Claudio Alfieri, who determined the structure of the APC-MCC and also the TRP13 PC31 comet complexes. The work on the APC was earlier work was done by Lifu Chang and also by Xuyang Sang, who determined the structure of the APC-C20 complex. And a lot of the biochemistry and molecular biology have been done by Sugu Sang and Ying Yang in my group. And then we've been helped by many of our colleagues at the LMB for computing and EM help and mass spectrometry, and also by physics, and also Carol Robinson and Florence Stengel, postdoc in her group, performed mass spectrometry on the yeast APC about 10 years ago now. So I'll finish here. Thank you for your attention.
Thank you very much for such a thorough talk on a complicated topic.
Very polite, thank you.
So David, I was just wondering, so the subunit needs to be phosphorylated for it to come out, right? So how does a kinase get access to
that's a very good question. Yes.
those sites?
Yes, I didn't explain that part of the study, actually. Yes. So that's the role of APC3. So I mentioned that APC3 and APC1 are hyperphosphorylated. APC3 is actually on the upper part of the APC, and the sites of which are phosphorylated are exposed to kinases. Whereas you point out, the sites which are phosphorylated in APC1 are occluded from phosphorylation by kinases. So what happens here is that CDK kinases phosphorylate the exposed phosphosites on APC3, and the CKS, which is a subunit of CDK cyclin CKS, binds to the phosphosites, and that actually then recruits the kinase to bind to APC. And then that positions the kinase bound to APC, which gives it more chance to phosphorylate this temporarily exposed phosphosites, because the inhibitory segment we found must occasionally obviously be released from that site, but that's probably quite transient. So to increase the probability of phosphorylation, if you localize the kinase to the APC, then you will actually increase the likelihood of phosphorylating those sites. If you delete the APC3 loop, which binds the kinase, you completely fail to phosphorylate.
And it's all the same kinase?
Well, the kinases are primarily CDK, but polar kinase synergizes with the CDK. So individually, polar kinase and CDK cannot fully activate APC, but together they do. So there's some priming phosphorylation involved by polar kinase, which allows CDK to phosphorylate the key sites which are involved in releasing the inhibition.
Well, my first question, Madame already asked. The second question is that, you know, the shape is really quite unusual, and you have lots of subunits on the left-hand side, but quite open on the right-hand side. Is that what's happening there? Is a substrate binding, or?
Yes, we've never been able to actually determine the structure of an intact substrate bound to the APC. So we try to, where we form complexes between the APC and the full-length cyclin, full-length CDK, CKS complex, and we could never really define very clear density for the full-length globular domains. We saw lots of, very diffuse density around actually APC3 on the front. So we don't exactly, I suspect actually that the APC doesn't need to bind the actual most of the substrate. It only has to recognize the degrons. Actually, those are the key. They provide all the affinity for APC recognition, actually. So that's why we can never localize the rest of the substrate bound to APC because it doesn't really localize a particular site. Yeah.
So, David, you explained very clearly a very complicated system, but periodically through your talk you had these beautiful fluorescence optical micrographs, movies, showing the chromosomes and, and the microtubules pulling them apart.
Yes.
So my question is there an equivalent, are there equivalent movies showing where APC is in that same movie, or APCs and kinetochores?
Yes, yes.
Because that would really, you know, bridge the gap between the beautiful optical microscopy and the beautiful molecular biology.
Yes, we are actually looking at that now. We are looking at the localization of the APC using super... We are now addressing that question Richard asked. So we are using super-resolution microscopy to determine the localization of the APC in situ. So it does bind to the catechores. It also binds to centrosomes, because centrosomes must also be divided prior to mitosis. They have to divide and then move to opposite poles of the cell. So the APC is regulating centrosome duplication or division, and also it's obviously controlling separation. So yes, the APC is localized to certain sites in the cell. We're trying to determine that. Eventually, we like to do by electron tomography, actually.
And do they appear at the right time, according to your explanation?
No, so the APC is probably consistently localized to catechores throughout the cell cycle, but it does appear to be dissociate from centrosomes during parts of the cell cycle. So yes, that would correlate with some of the known functions of the APC at centrosomes.
Well, if there are no more questions, let's thank David for a beautiful talk.